Good morning, everybody. Welcome to the RCN webinar. We're going to get started in a couple minutes as everybody trickles in. Okay, we're going to go ahead and get started. First, I want to introduce everyone to Sarah Philo, who is the new postdoc working at the RCN. So you may be getting emails for her from her. And I want to introduce our two speakers today. The first speaker will be Janelle Thompson, who is a professor at NTU. And the second speaker is Tyler Radniecki, who is a professor at OSU. And we're really thankful for these speakers to having agreed to speak with us today. And I have to give a special shout out to Janelle, who is in Singapore, where it is midnight right now. So thank you so much for being willing to not go to bed yet and stay up to talk to us about your exciting work. Also, the second announcement is just a reminder of our next webinar in April, which will feature two prominent public health specialists who have really been at the forefront of using wastewater-based epidemiology, and we'll be able to hear about, where they are now in terms of using wastewater and where they see this going in the future. And the third and final announcement is that if you would like to nominate a speaker for next year's webinar series or have any other suggestions for content, please scan that QR code and you can fill out the information online. So I'm going to go ahead and hand it off to Janelle. While she's sharing her slides, I want to remind you all to put your questions in the Q&A, please, and I will be asking those questions to Janelle at the end. Okay, Janelle, thank you.
Great. Well, hi, everybody. It's really a pleasure to be here speaking to this network of people who are doing really important work. As Ali said, my name's Janelle Thompson, and I'm based here in Singapore. So on the other side of the world from most of you all. I'm based here in Singapore at the Nanyang Technological University. I am an associate professor at the Asian School of the Environment here and a principal investigator at CELSI, the Singapore Center for Environmental Life Sciences Engineering. So during the pandemic, I've been co-leading a collaborative research group working on wastewater surveillance, both for SARS-CoV-2 as well as for some emerging pathogens and endemic pathogens here in Singapore. So I hope to tell you a little bit about both of those activities. And my key collaborators on this work would be Stefan Wirtz here at Singapore, the Singapore Center for Environmental Life Sciences Engineering, and Eric Alm at MIT. And I hope that you've just noticed already that my brain is a little bit, I have a little bit of a case of midnight brain. So please bear with me, and please have good humor as we go through this. All right, so I'm excited to tell you about what we've been working on, and let me advance my slides. So we're preaching to the choir when we talk about wastewater surveillance and its importance. We can use wastewater surveillance both to look at the spatial distribution of a pathogen as well as its temporal dynamics. And so to that end, we're thinking about sampling both at maintenance holes in neighborhoods and on university campuses, as well as downstream at the wastewater treatment plant. And then we can use that information for various policy implementations. So the work I'm going to tell you about today pertains to our use of wastewater surveillance that provides situational awareness for endemic COVID-19. And this is becoming increasingly important as the magnitude of clinical reporting declines, right? Now, we're also maintaining vigilance for emerging variants of concern, and we see wastewater surveillance now, all this infrastructure that we have on our university campus, that many people have on their university campuses, as a platform for surveillance of new pathogen targets. So this is just some data from our university campus monitoring program, juxtaposed against monitoring that we've done at Singapore's wastewater treatment plants. And so you can see here, the red line is a smoothed average of the SARS-CoV-2 levels through the Delta and first part of the Omicron wave at Singapore's wastewater reclamation plants. And then the black lines here are the proportion of campus sites where we've seen positive signal for SARS-CoV-2. And we've been monitoring 45 sites through the pandemic. And this is work that's been supported by Singapore's National Environmental Agency, and it's really part of a nationwide monitoring effort that the NEA coordinates. And so you can see, the nice alignment here between the trends that we see in terms of concentration at the wastewater reclamation plants and what we're seeing on campus. While we've been doing this work, this routine monitoring, we've also been developing assays to track variants of concern. And so a lot of the heavy lifting on this work has been led by Dr. Wei-Lin Lee, who you see pictured here. And these assays target mutations in the spike proteins. So these are these little external proteins on the SARS-CoV-2 particle. And so we've developed assays that target the alpha variant, or mutations associated with the alpha variant, delta variant, a number of different variants, as well as Omicron, and then its sublineages BA1 and BA2. And these are allele-specific PCRs that target a specific mutation, and we have developed paired assays. So one assay will target the wild type version of the sequence, and the other assay will target the mutant version of the sequence. And so putting those two together, we can determine the dynamics of these variants and their sublineages in a sample, and we're focusing on wastewater samples. So we've done this over the course of the Delta and Omicron wave, and this work has been done by postdoc at CELSI, Desmond Chua here. And what we see is the kind of clear mapping of the Delta variant of concern during this first peak between September and November in 2021, followed by a period of quiescence where we didn't have a lot of circulating SARS-CoV-2, and then boom, we had the Omicron wave. So one of the things we were wondering in Singapore was whether we saw dynamics of BA1 followed by BA2, which had been reported in a number of other places around the world. And in fact, Singapore had a number of policy changes during this time, which kind of coincided with nationwide vaccination campaigns. So you can see here on this timeline, on the bottom plot here, how our vaccination rates have steadily climbed through the months. And by the end of December 2021, we reached a target of about 85% of our population being fully vaccinated. And concordant with this time, the government relaxed some pretty significant policies enabling easier travel in and out of Singapore, and just a little bit less of a stringent lifestyle. And so upon that relaxation, then we saw the rise of the Omicron wave. So when the Omicron wave began, we actually saw a coincident detection of both variants, both BA1 and BA2. And so this contrasts places trends that were observed elsewhere, where BA1 first emerged and then BA2 came in and displaced BA1. So right at the beginning of the Omicron wave, we saw co-occurrence of both BA1 and BA2, but we saw rapid dominance of BA2 by early March. Now, these are trends that we observed at the wastewater reclamation plant. If we look at our campus, We see qualitatively similar results, but again, with the campus, it's a much smaller catchment size. And so it's to be expected that you see quite a bit more variability there. So hopefully this gives you a little bit of a sense of what we've been doing. You know, we've been maintaining situational awareness for our university campus as part of this nationwide monitoring program, but we've also been interested in following the evolution and emergence of these variants. We've developed protocols to reasonably rapidly develop these assays that can target high-risk pathogen groups. And this is important because now that we've moved into a period of endemic COVID-19, we're maintaining situational awareness and we're poised to respond if a new variant should emerge with a risk profile that is much more alarming. So the second direction of work that we've been pursuing is thinking about developing a wastewater surveillance for arboviruses. Now, what's an arbovirus? So it's a kind of polyphyletic group of organisms that are named based on the fact that they infect arthropod vectors. So it's arboviruses. Most of the arboviruses, or many of them at least, are flaviviruses. So they're all related, but there are also some arboviruses that are not flaviviruses. Okay, so arboviruses are concentrated in the tropics in alignment with their vectors. We also expect that there's an increased geographical spread of these vectors with climate change, and thus that will spread further these arboviral infections. And interestingly, like COVID-19, many of the infections of these arboviral pathogens are mild or asymptomatic. And in fact, it's estimated that 50 to 80% of these infections can be asymptomatic based on seroepidemiology. So a few examples, we have dengue, which causes about 96 million cases per year. Estimated about 75% of those are asymptomatic. And of course, there's a large healthcare cost associated with that. Yellow fever is another important arbovirus, primarily in South America and Africa, causes significant morbidity and mortality. West Nile virus influences regions all around the world, and then Zika virus as well is an important arbovirus. Now, currently, arboviral surveillance centers around three main pillars, looking at surveys of the animals that are kind of the primary host for these arboviruses. These would be the sylvatic surveys, then entomological surveys where you check whether mosquitoes are infected, and then surveillance that are really focused on human populations. And this surveillance pillar focuses on clinical reporting. The problem here is that clinical reporting, of course, misses the asymptomatic and subclinical majority. And so what we've been thinking about is the fact that we could use wastewater surveillance to get better insight into the clusters that may be primarily asymptomatic. So we want to ask basically, could wastewater surveillance be used to help track arboviral outbreaks? So to look at this, we've considered shedding rates of arboviruses, whether arboviruses can persist in wastewater, and then whether we can recover them from wastewater. So this is a figure from a review that we put out recently that shows different levels of arboviral shedding in relation to some kind of well-established wastewater surveillance targets. So here we have SARS-CoV-2 in black. The y-axis here is the RNA copies per day that are shed per infected person. So we see polio, which is a long-standing target, one of the longest standing targets of wastewater surveillance, and then norovirus here. And then here in green, we have some urinary shed pathogens. You can see their distribution in terms of shedding rates. And then we have a relatively limited data set on shedding rates, urinary shedding primarily, of dengue, West Nile virus, Zika, and yellow fever virus. And so what you see here is that the published data on shedding rates is rather limited. It's also highly variable. And urinary shedding does seem to be the primary route, although there are alternate routes of shedding as well. The median rate of shedding for these arboviral targets is lower than the fecal shed pathogens and somewhat comparable to the other urinary shed pathogens in this review. So we did some back of the envelope calculations to see whether enough arboviral signatures would be shed in wastewater to be picked up during the sort of testing that we do. And so we considered different volumes of wastewater processed. In our campus network, we routinely process about 50 mils per sample. We considered a case where you scale that up and process 500 mils, which is feasible in our testing setup. So if we're testing 500 mils of wastewater, we estimate that the detectable incidence would range from one infected person in a population of 50 people to 2,500 people, considering the median shedding rates for those arboviral targets. And in the optimistic scenario, one infected person in about 500,000 people with the maximum shedding statistics used. And so the take home from this back of the envelope calculation here is that we're really going to need larger volumes of water of sample to be able to detect these arboviral targets relative to SARS-CoV-2, which had those higher shedding rates. So we also asked whether arboviral targets can persist in wastewater long enough for us to detect it, and thinking particularly about the time frame for forming a composite sample. So our composite samples are 24-hour composites. We collect our composites under tropical conditions. So will the tropical condition impact the persistence? And what about pasteurization, which we commonly use to increase the biosafety of our method? So Francis, who you saw on this slide here, he carried out a body of really nice work where he set up these wastewater microcosms with these different viral targets. So we actually used yellow fever virus, Zika virus, dengue, and murine hepatitis virus as a control. Incubated them at the temperatures indicated here. So 37°C, that's our tropical condition, 25, and then 6 degrees C, so a refrigerated autosampler condition. And then we followed their decay in wastewater that was collected from a building in Singapore. And we saw that about 90% of the signal was lost between 3.7 and 6.7 days at 25 degrees C. Not surprisingly, the signal decayed even more fast at 37 degrees C, between 2.4 and 5 days. And we observed that the signals would be stable for over 21 days at 6 degrees C. We also saw that pasteurization was okay. So let's put that figure here, that we didn't see any loss of signal when the samples were processed right away after pasteurization. So in this plot here, the black bars represent pasteurized samples, and the red bars are the completely raw samples, and you can see similar rates of viral target detection in that. So that provides some assurance that the biosafety protocols can be used. All right, so we do see significant decay, but nevertheless, we can still, after 1 day, pick up enough sample for this to be a viable approach. And last, we wanted to see if we can recover environmentally relevant concentrations of the arboviral targets. And so again, Francis carried out a second study where he evaluated different methods of sample clarification, the filtration or centrifugation, different methods of viral concentration. So these are the 4 here: ultrafiltration, PEG precipitation, hollow membrane, hollow fiber ultrafiltration, and charged membrane capture and elution. And lastly, looked at different viral nucleic acid extraction methods. So we evaluated these steps separately in wastewater with spiked virus, and then combined the approaches to test detection at environmentally relevant levels. And so here you see, for example, this is a 50% recovery of viral signal using centrifugal ultrafiltration in contrast to some of these other methods. So that was the best performing method, was centrifugal ultrafiltration. Best performing for sample clarification was using the filtration. So putting these pieces together, we did a proof of concept where we showed that at concentrations that were equivalent to between 1 in 40 people to 1 in 40,000 people, we were able to detect the DNA concentration or the RNA concentration corresponding to a single case. Now, this is very much for illustration purposes because we had to take note that there's just such high uncertainty in the shedding rates for these targets. So this is really for illustration purposes to show, though, that it could potentially be feasible to use this approach to detect dengue at the scale of a cluster in an apartment building, or possibly yellow fever in a small township. And I can say that this study was accepted in ES&T Water, and so that is in press at the moment. So hopefully that'll be out soon so that you guys can check it out if you're interested. So to wrap up then, so the implications and our future work for endemic pathogens. So first off, endemic COVID. We're maintaining vigilance for high-risk SARS-CoV-2 variants. We're continuing our routine surveillance and then standing by with capabilities to track new variants of concern, God forbid if the need arises. We're also working to optimize arboviral surveillance. And we've seen that arboviruses are shed in urine at levels predicted to be detectable by wastewater surveillance. But we really do see that the shedding data is very scarce, and obviously our predictions will significantly improve with better data. So we've demonstrated that arboviral signatures can persist in wastewater long enough for sampling, so longer than 2 days, and that they're compatible with pasteurization for safety. And also importantly, that refrigeration significantly improves signal stability. So that's an added suggestion to get a auto sampler with refrigeration capabilities for us at least. So we think that there are arboviral targets that have higher shedding rates that actually might be the best targets for a wastewater surveillance approach, such as yellow fever and Zika. And so we're still working on this. We're looking at ultrafiltration systems that support higher input volumes, as well as quantifying the impact of viral sorption onto particles in our community samples. So this is an area that we're working on, and I invite you to stay tuned to further developments. Lastly, I want to thank my research team and key collaborators. We've got Stefan Wirtz here, here's Eric here's Waylin, Francis, this is Desmond, Jason. So we've got a wonderful collaborative team, good friendships, and yeah, it's certainly been real during the COVID-19 pandemic, but it's great to have good people to work with. So with that, I'd like to wrap up and take any questions. Thank you.
Thank you, Janelle, for that great presentation. And I could not tell that you had midnight brain, so I thought it was fine. So thanks a lot. Okay, so please put your questions in the Q&A, and I'll ask a question or two while we get more questions there. So my first question for you, I think is related to a question in the chat, and that's about the persistence of the viral RNA. So, I mean, like I always was told like RNA is really unstable in the environment. And I've always been surprised that these small fragments of RNA that we target with our qPCR and digital PCR are so persistent. And I was wondering if you had any thoughts about do we need to keep testing every, all viral RNA that we target for its persistence? Or do you think, do you have an idea of the mechanism whereby, RNA is degraded? Are all small fragments that are like 100 base pairs going to persist at the same rate? Do you have any thoughts about that from your molecular biology training that you can share?
Well, I think the idea that that RNA isn't stable comes from studies with mRNA. And viral RNA is not mRNA, right? So viral RNA has crazy amounts of secondary structure, and it's often protected in a viral particle. Now, if it's fragmented RNA, just like free floating in the wastewater, that could be more like mRNA, right? But to the extent that it's in a viral particle, I don't think it's too surprising that these things can be crazy persistent.
Yeah, that makes sense. And I think that segues nicely to a question in the chat, which is sort of commenting on how the persistence of the spiked viruses versus like endogenous native virus might vary, because the spiked virus could remain intact, but the native virus could actually be like fragmented or broken in some way. What are your thoughts on that?
I don't think I understand the question. I'm sorry, maybe I'm not a good enough virologist to get
I think the question is like, when you spiked your virus in,
okay, I'm thinking spike protein. Okay,
no, sorry. The added virus that you added was probably intact and might be different than the native virus, or in both, like, you know, how like maybe the native virus is broken, or just, you know, outside the capsid, the RNA is outside the capsid, or maybe it's like coated with a bunch of like organic carbon and that, you know, that sort of.
Absolutely. So when you do a spike experiment, I mean, there's always bottle effects. And I mean, a spiked virus was grown in a culture. It wasn't like subjected to the human gut, right? And so it doesn't have the same sort of surface modifications it would have had having gone through. So really, I mean, there's a significant amount of uncertainty attached to these sort of experimental works. And so it's a proof of concept, but ultimately, you know, the question is, can you detect it in the field, right? Mm-hmm. And so Ali, you and I have talked before, I mean, we haven't yet detected dengue in the field, and you know, knock on wood, we don't have the opportunity to test yellow fever, or Zika in Singapore, but dengue is endemic here, and we haven't recovered it. We haven't detected it yet at high enough concentrations, at least. So yeah, absolutely. There's a lot of uncertainty in all of this, and people are very wise to keep that in mind.
Great. Okay, so that now there's two questions in the chat that are really related to each other. One is from Marlene and one is from Alessandro, and they both relate to partitioning to solids. And so Alessandro says, you know, a lot of the literature seems to suggest that arboviruses are shed in urine versus feces. Did you find any reason to believe that the viruses would remain in the liquid phase and not partition into the solids like SARS-CoV-2, influenza, etc.? And Marlene's question is very similar. Yeah.
Right. So we have some preliminary data where we did the spike and looked for partitioning. And it seems like it does partition into the solid phase. So that's originally why we targeted the supernatant, was because it was shed in the urine, right? But in an autosampler, it's hanging out with particulates for 24 hours, right? Or at least 24 hours. And so that was the model system that we used in our preliminary experiment. And we definitely saw partitioning into the solid phase. Now that's a one-off. I mean, we haven't followed up with that yet to really get the kind of partition statistics yet, but it did seem that it was about 50-50 at the end of 24 hours.
Okay, interesting. All right, we have time for
I think there's really good questions there.
Yeah, we have time for one more question. And then Janelle, if you want, you can answer questions in the Q&A if you'd like to, the remaining ones. So the last question we have is from Liz Mercier, and she asked, do you have any thoughts on what is the species, meaning form, I think, of RNA shed into wastewater? Is it messenger RNA? Is it viral RNA? Is it protein protected? We kind of touched on this a little bit, but do you want to add any thoughts to that?
I mean, we test the scenario that it's viral particles, and I think there's data to support that, but it could absolutely be RNA fragments. Is it message? I mean, I guess we sort of went through the figure, like, so my expertise is not on how gastrointestinal epithelial cells respond to viral infection, but we did kind of walk through a figure at one point and thought, you know, it is possible that there is mRNA that's being produced by this. So the answer is, I don't know. And it would be great if somebody who had the expertise could actually figure that out and give us a really nice conceptual model for it.
Yes.
It's a great question.
Thank you so much, Janelle. We are going to move on to our next speaker.
Thank you for having me. It was very nice to be here. I'll look at the Q&A questions. Bye-bye.
Bye. So our next speaker is going to be Tyler, who will share his screen. Janelle, are you able to stop sharing? Thank you. And Sarah will moderate this talk. Tyler, you're muted. Oh,
Sorry about that. All right, thank you, Allie and Sarah, for the invitation to speak to everyone today. Today, I want to tell you about a project that we did last summer, where we were using wastewater surveillance to identify the potential public health impacts of large international events on host communities. I need to come up with shorter titles, but that's where we're at. So as a research community, I think we've had a very valid discussion about where does wastewater surveillance have the most impact, right? There's been many discussions about when and where should we do it. And I think most of us will agree that it has the most impact when the clinical data is the most sparse. And so here in Oregon, we've demonstrated this with our statewide surveillance program that we've done. Here's a map of Oregon. The red circles are cities in which we were detecting the Omicron variant. This would have been back in December of 2021 when it was first coming to the state. And in the shaded green you see there, that is the percent of clinical samples that also came up positive for Omicron. And so you can see early on when we first detected Omicron in the state, from a wastewater surveillance perspective, we saw it in quite a few communities up and down the Willamette Valley and the I-5 corridor, as well as out in Eastern Oregon along I-80. But with clinical samples, they were only seeing it up and down the I-5 corridor, which is not surprising because that's where most of the sequences happen. About 80% of the population lives along this skinny strip of land. And this is also where we have our three major universities: Oregon Health and Science University up in Portland, Oregon State University in Corvallis, and the University of Oregon in Eugene. All three universities were sequencing clinical samples at this time in their regions. But as we monitored over time, we could see we start detecting the Omicron variant in the wastewater more and more, and we're outpacing where we're seeing it in clinical samples. And by the end, here now we're getting into January of 2022, you can see by the end of this whole surveillance, we detected Omicron in the wastewater prior to clinical in 12 counties, all of which were outside of that tight surveillance zone of the I-5 corridor, north and south, from Portland to California. In fact, you can see on this last map, there's still some counties that, clinically speaking, they still didn't think they had Omicron present, where we were showing it was by far the dominant variant we were seeing in the wastewater. And so the sparsity of clinical data surveillance data, can be both spatial. So this, our example here, we had rural areas of Oregon versus, again, the I-5 corridor. It can be temporal, right? So clinical surveillance efforts will drop as funding ebbs. During the COVID-19 pandemic, that's probably the best clinical surveillance data you can probably possibly have. And we're already seeing it starting to get more and more sparse. But it can also be used for transient events, such as sporting and cultural events, tourism, where you have people coming in and coming out that aren't captured by the local clinical surveillance network. And that brings us to the World Athletic Championships. So the World Athletic Championships are hosted in Eugene, Oregon. From July 15th to July 24th in 2022, last summer, it was a very exciting time. The entire state of Oregon was very proud to have this major event being hosted at the University of Oregon campus. And what you can see was that it was held in Eugene, Oregon, population of about 235,000. There was 150,000 ticketed spectators from all 50 states and 40 countries. There were 17,000 athletes from 180 countries and 3,000 members of the global media, all come into this reasonably small community. And so while the community was extremely excited to have this influx of individuals to their city, they were also very nervous about what types of diseases might these visitors be bringing from around the world. For those of us that have done campus wastewater surveillance, this isn't that dissimilar than the local host city being very nervous about campus opening up and having all the students come back to campus, and what are they bringing in terms of COVID and other diseases. There's been a heightened awareness around this topic. And so to that end, we conducted a wastewater surveillance program for the World Athletic Championships, which we're going to call the WAC. We selected 8 locations throughout the city. We have one manhole here, which we're calling pre-campus. This is collecting the wastewater entering into the WAC. We had 2 on-campus locations where the WAC was being held. We had a post-campus, so this collected all the wastewaters that left the university. All of this flowed then directly into the wastewater treatment plant, which we were also surveilling. We also conducted surveillance on 3 hotels located close by where many WAC spectators and participants were staying, as well as the Eugene Airport. We had a manhole out there that collected wastewater just from the airport. Again, we collected 24-hour composites from both the treatment plant and from those selected manholes. We filter our wastewater samples through electronegative membrane filter. We then extract it, use ddPCR, as well as sequence results, and then send that to the authorities. In this case, the Lane County Public Health Authority, as well as the Oregon Health Authority. As far as the pathogens, this was an opportunity for us to expand our portfolio of the type of pathogens that we look for. We were already looking for SARS-CoV-2 and influenza A and B across the state, but we are also, there was a lot of concern about adenovirus, 4041, hepatitis, both A and E, as well as MERS and measles. So these were the 8 pathogens that we targeted. And throughout the event, we only detected these 3. SARS-CoV-2, influenza A, and then adenovirus 4041. So let's quickly talk a little bit about the SARS-CoV-2 results. So this is typical time series data. We have log 10 of the gene copies of SARS-CoV-2 on the y-axis over time. The blue shaded regions is the WAC event itself. So you can see we took samples both prior to WAC as well as after WAC, and then real intense sampling on a daily basis during the WAC. And what you can see from the SARS-CoV-2 data shown here is that the wastewater treatment plant really didn't change too much in its viral load over time. It stayed more or less consistent. It was a little more variable, but not a lot of changes. Interestingly, at the airport, we saw a marked decrease, including even a non-detect one day. And that caught us a little bit by surprise, but maybe it shouldn't have when you think of the higher percentage of international visitors coming through this airport and the reasonably strict COVID regulations that was on international airline travel at that time into the United States. And so in many ways, it was actually the lowest abundance that we saw in our surveillance. But meanwhile, at both the hotels as well as on campus, both pre and post during the WAC event, we saw a spike. We saw an increase in SARS-CoV-2, especially pre-campus. That we basically detected no COVID prior to the WAC and no COVID essentially after the WAC, but we started to get this spike during the WAC event itself. On campus and post-campus, they mirror each other, where you saw lower levels before the event and after the event, but during the event, we had heightened levels of SARS-CoV-2. So this was telling us that we were getting localized increases of viral shedding within the community that surrounded around the WAC event at the WAC site. If we move on to influenza A, again, very similar charts I'm showing you here, gene copies per liter of influenza A over time. We saw at the wastewater treatment plant, we only detected it one time and at a very low level, just above detection limits, which was telling us that it wasn't extremely sensitive at the wastewater treatment plant. It makes sense that we weren't seeing it before or after because this is the off season for flu in Oregon at this time. But we did get one little blip during the event. You can see though, everywhere else we looked, we definitely saw spikes, especially on campus, post campus. We had one spike at the airport and a little bit at the hotels that occurred during the WAC. And so this was telling us that we were getting an influx of influenza A into the community at reasonably low levels. We didn't see it too much at the treatment plant, but we were seeing it localized where we had higher sensitivity. Interestingly, you'll notice that there was a residual spike after the WAC event and roughly 8 days to 2 weeks after, which happens to be the incubation time roughly for flu A. And so our epidemiologists, when they were looking at this data, they're like, oh, that looks very interesting to us. We think what's happened here was we brought flu A to Eugene through the WAC participants and spectators, and it stayed past the WAC for essentially one cycle, where you can see little mini flu outbreaks, which then died off after another cycle or so. Importantly, during this whole time, there were no cases of clinical influenza being reported. So all of this influenza bringing in, staying, and leaving would have been completely blind to the public health surveillance if it wasn't for having this wastewater data. And this was very valuable information to our public health authorities. Finally, I'd like to talk a little bit about adenovirus 4041. Very similar chart shown here. Perhaps the most interesting thing we found with adenovirus was that it was everywhere. And that kind of surprised us. Everywhere we looked, we saw reasonably high, well, what we believe to be reasonably high concentrations of adenovirus, particularly at the wastewater treatment plant. So it wasn't even like the WAC event was the significant source of adenovirus, because everywhere you look that we associate with the WAC, we're about an order to 2 orders of magnitude lower on the adenovirus than what we see at the wastewater treatment plant. So this is telling us that the adenovirus is actually quite endemic, at least in the Eugene community. And that, I think, surprised a lot of our public health collaborators. And the reason this became of a higher concern is because adenovirus, there's been some studies coming out in the last year where they're starting to link it with acute hepatitis and liver failure in kids. But now the question becomes, a child goes in for acute hepatitis and liver issues, and they test them for the gamut of viruses, and they say, oh, you have adenovirus. So it's like, oh my gosh, we're getting all this adenovirus whenever we're looking at kids that have hepatitis in the hospital, liver issues, terrible things like that. But the question is that adenovirus really being linked to that hepatitis and liver issues? Or is it, if you look at healthy kids, which you see equivalent levels of adenovirus? And the wastewater surveillance is already starting to suggest that adenovirus is much, much, much more prevalent than people might realize. And so it may not actually be causative at all, just correlative in this hepatitis and liver failure that they're seeing in pediatric cases. And so in conclusion, wastewater surveillance was useful for evaluating the health of transient populations. In this case, our WAC spectators, athletes, media, staff. We were able to see that additional SARS-CoV-2 was brought into the community, but because we didn't see it at very high levels, in fact, lower levels than normal at the airport, which is where we're assuming most of our international visitors were entering into the community, we're thinking that primarily this increase of SARS-CoV-2 was brought in from domestic sources, from other Oregonians and people in the region and the country that came to Oregon to watch these events. I apologize, I didn't show the data, but we also did sequencing of all of our SARS-CoV-2 positives. And when you look at the subvariants of Omicron, so BA2, BA4, 5, 7, I think we even had BQ1 and BF7, you didn't see any real significant differences in the relative abundance of those subvariants, saying that we had a pretty consistent input of SARS-CoV-2. And we were hypothesizing if we had a lot of COVID coming in from international sources where different variants were dominant at the time, we would start to see these fluctuations in the variants of SARS-CoV-2, but that was not the case. But the WAC did bring off-season influenza. This we are currently right now hypothesizing came from international sources because there were no clinically reported cases of influenza in Oregon at this time. It's the traditional off-season, but yet we did see it come during the WAC. And then we even saw it stay for 1 to 2 incubation periods after the WAC before it died out again. And this can be extremely important if you had viruses or diseases that were even more contagious or had even more serious outcomes to influenza coming to your community. If you have lots of visitors that aren't going to be a part of your traditional clinical surveillance system, having a wastewater system set up will at least allow you to understand if that virus or that disease is coming into the community and can help you keep an eye out for it. And finally, we saw that adenovirus was much more widespread than many assumed. And again, this becomes important as people are starting to try to link adenovirus in kids to other more severe diseases. The question is, how widespread is adenovirus in the community? And I think wastewater can play a big role in understanding just that phenomenon. Just, is it common everywhere, or is it really only in select locations? And so with that, I have a whole large host of people to thank, but in particular, I want to point out Dr. Christine Kelly, who is a professor of bioengineering here at OSU and my collaborator in all things wastewater. My postdoc, Devram Kaya. Michael Harry was our field technician. He was the one that went out and set up all the wastewater samplers. He was the one that interacted with the FBI when they came by wondering why we had battery operated large devices in wastewater sewers. That was interesting conversations. As well as our Oregon Health Authority, Doctors Melissa Sutton and Doctors Paul Cieslak, Noah Lindinger, Ernesto Rodriguez, and Dr. Rebecca Fallender, and our Lane County collaborators, Dr. Patrick Lutke, who's the Public County Health Authority lead there, the City of Eugene for providing us access to all the manholes and the maps, and the University of Oregon for giving us access to their campus. And if there's any time, I'd be happy to answer questions.
Great, thank you so much, Tyler. It was very interesting. So one question that's been posed in the Q&A is, did you consider normalizing any of your data for flow or PMMOV or population? It seems that flow would have been much higher, particularly on campus in the summer, than you would have expected normally. Yeah,
that's a fantastic question. So at the wastewater treatment plant, as part of our routine surveillance, we do normalize everything to flow. Where it gets trickier is in the manholes, and to be honest, we just don't have the equipment. It's quite spendy. Those of you that have bought, $5,000, $10,000, depending what type of flow meter you want. So that got tricky. We did measure BOD as a way to try to normalize that in that way. The BOD was actually quite consistent, which makes sense because really the flow should increase proportionally with the number of people. This is Oregon in the summertime, so very Mediterranean climate, so no rain. And so the flow in the wastewater is really directly correlated to the number of people, running faucets and flushing toilets. And so the relative concentration stayed, roughly the same throughout. And then as far as the number of people, again, with the influx of visitors, that became a very open question of how many do we have? And on campus, we had some idea, but once you start getting in the hotels and all the Airbnbs in the area and things like that, it got much more challenging to estimate what the populations were. But yeah, that would be fantastic. That was definitely a limitation.
Yeah, I wonder if it would be similar to like football games on campus, just population size. I know campuses get a lot of people around those college games. Yeah.
I think it would be analogous to something like that.
Yeah. And so then there's also some questions about the non-detects for measles and then the hepatitis viruses. So how did you interpret that? And then are there plans for publishing any of those non-detect results? Just because that's also a very interesting piece of this.
Yeah, no, absolutely. So I was trying to keep my talk reasonably short, so I didn't focus on the non-detects. So for all of our assays, we run the full panel of what's our LOD and LOQs. And so non-detects, in our case, so we are doing droplet digital PCR, and internally we say we must see at least 3 positive droplets out of 20,000 plus to make a positive call. That also corresponds well with our LODs and LOQs that we see for these different targets. So the fact that we didn't see measles or hepatitis on the one hand is very good from a public health standpoint. On the other, we will publish our results. We'll publish, we're working on the paper now, the manuscript now. We'll publish the negative results as well as our LOQs and LODs so people can get an understanding of at least what we were capable of doing. And I should mention that, this was done in a reasonably short period of time. I think we had about 2 months to get most of these assays up and ready because this event just kind of got thrown on us, and the health authorities said, hey, why don't you do this? We said, sure. And we just did the best we could in a reasonably limited amount of time.
Yeah, great. And then there's one question about if you had detected measles, like what would the response for that have been? And then sort of a follow-up to that, did public health, with the flu data, did they do any sort of like messaging campaigns? What was the actual response to the data that you did have? And then was there a plan for what would have happened if you had detected something like measles, for example? Yep.
No, those are also really good questions. So part of this was a pilot project. Okay, so it wasn't linked into a rigorous outreach program. So this was funded by the state as a pilot of, can we do this? So when we detected the influenza, everyone found that really interesting, but there wasn't the machinery in place for a real-time response this time around. Now that said, for measles, because we knew how serious that would be, we did strategize, game plan how you would respond to a positive measles. And so if that was to happen, first we would confirm our results. And once confirmed, it goes straight to the head of the Oregon Health Authority, as well as the governor's office, and they make statements to the communities. And then from there, I believe the public county health authorities, so Lane County Public Health, would start to take additional steps in the areas where we saw measles. So we saw it located just on campus, or we saw it located just at one hotel. They'd probably go in and do additional outreach. I don't know if they would have done surveillance. This is outside of my league. I just measure the wastewater, but they were ready to act on that one because that one was such a significant concern. But fortunately, we had all non-detects throughout our study.
Yeah, that's always good. And then there's a question about both pre-campus flu detects, but also the other viruses. Maybe you could talk a little bit about where some of those like Airbnbs would be, other communities and stuff.
Yeah, I should have explained that a little better. So if for those of you that have been to the University of Oregon campus or downtown Eugene, so it's a pretty typical college town as far as you have the campus itself with its dorms, but then you can basically make a perimeter around the campus that's almost all rentals, college students, graduate students, for the most part. And so during the WAC, a lot of students I know were making a lot of extra money renting out their apartments for a month, and which was great for them. So I wasn't extremely surprised that we saw, for instance, flu pop up pre-campus, even though you would have thought, well, that should be the rest of the Eugene Springfield community, the metro area coming into campus. But right around there, before you enter campus, that's a heavy rental area. So I'm sure there were many sub rentals going on to visitors to the WAC. Same thing with post-campus. Again, that's kind of a graduate student, undergraduate rental type of neighborhood.
Great. And then in terms of that, like validating the results, did you do any sequencing on the flu or the COVID, like what was the process to actually validate that your ddPCR results were correct?
Correct. So we didn't do any sequencing outside of COVID. So we did sequence all of our COVID results. And like I said, we were getting variant distributions that matched what we were seeing in the community before WAC, after WAC. So there was nothing really out of the ordinary. It matched what we were seeing across the state. We monitor about 40 communities across the state, and it looked about the same. As far as our other assays for confirmation, I mean, we did develop G blocks for each of the different assays, right? So we knew it would amplify. So for instance, influenza, we actually, I take that back. For influenza, we used a CDC standard, which had 9 different strains of influenza. And so we knew we had positive controls. Now, the question might be, do you know if you had false positives or not? And that one I cannot answer at this time.
Okay, and then I think we have time for just a few more questions. Did you have to change the methods at all for the DNA versus RNA targets, or did you sort of do the same workflow? And then there's also a question in the chat about just talking generally about your lab workflows for sequencing and analysis.
As far as the specificity between the RNA targets and the DNA targets, we did have a lot of questions on that. Like, would we need to do? We were doing the same concentration method, but the question would be, would we have to split, do additional filtrations and split samples, do RNA extraction on one and DNA extraction on another? And we did some of that early on just to see, and it turns out, at least our RNA kit that we're using happens to also do a pretty good job of getting DNA out as well. I don't know if it's supposed to, but it does. And so we were able just to have a single workflow all the way through and felt reasonably comfortable with our results from that, because we did do some cross studies of RNA extraction only, DNA extraction only, and then in the RNA extraction, look for DNA targets, and they came out reasonably similar. As far as the larger workflow, I guess I'm not quite sure how specific this individual would like it to be. I'm happy to answer in more details, though.
Yeah, maybe they can follow up with you on email, I guess.
Yeah, I'd be very happy to do that. Absolutely.
And then I just have a sort of a general question. Do you think that this is a useful thing moving forward with large sports events? I'm thinking mostly about the World Cup being here. Like, do you think that it provided utility, at least to public health, for figuring out what was going on?
Yeah, I do. I think our county and state public health authorities, I think they were quite pleased with the data. One of the challenges, especially in these fleeting events, so to speak, is you got to get data out extremely fast, and you got to have high confidence in the data that you do get out. And so that is always going to be your challenge. If, for instance, if the World Cup was coming and people are looking, you might want to target what are positive detects where if we see this, action must be taken now, polio, measles. I let the public health authorities decide what's the most pressing, vital ones where immediate action must be taken, versus others where it's like, oh my gosh, we're getting this big influx of flu and we're seeing it. So let's let the local hospitals know that there's a chance of flu outbreaks, be prepared, be stocked, be staffed. I think that's another big one, is using wastewater data to help the local responses be prepared, right? And I think that has a lot of value.
Great. Well, I think we are all out of time. Thank you both, Tyler and Janelle, for really interesting, wonderful talks and question and answer. Janelle for staying up very, very late. Yeah. So,
thank you, everyone. See you next month. Bye.
Thank you, everybody. Appreciate it.